It is customary at this university to start the ceremony with a prayer, with some words. Spiritus Sancti Gratia Illuminet Sensus et Corda Nostra. You may be seated. Good afternoon, ladies and gentlemen, and welcome to this academic ceremony in which Lynn Lee will defend her dissertation. My name is Joukje Oosterman. I'm a professor in neuropsychology at this university, and I will be acting rector today. Because of the international composition, this session will be held in English. I would like to address a special word welcome to the members of the promotion committee who traveled from their home universities to be present here today and participate in the ceremony. Thank you very much. I now give the floor to the candidate of the degree of doctor.
With the permission of the doctorate board and in order to obtain the degree of doctor from Radboud University Nijmegen, I would like to defend in public my dissertation entitled Reconstructing Visual Experiences from the Neural Code with Deep Learning. I will walk you through my PhD journey and show you what I have been working on for the past years. And basically, I did a little bit of mind reading with artificial intelligence, and I will explain why and how. So, vision. When we look around us, we process visual information constantly, and light falls on an object, and this visual information goes into our eyes, and the eyes sends it to our brains, and the brain somehow makes a meaningful perception of or representation of the world around us. And we do this without even trying our best or even thinking about how to do it. It just, our brain does it. And it seems quite effortless, but it is quite a difficult or complex task for us to understand how the brain does it. So when we try to build a system that is able to interpret images the same way as we do, this is called computer vision. We first have to train it and then test it, and then there might even be a chance that it's not even as accurate as the brain. So computer vision costs a lot of energy as well, so a lot of computational resources. So it's not very efficient. Our brain, on the other hand, is very efficient. It only uses the same amount of a light bulb to do all of these computations. And that is why I have always been inspired by the brain. And in my research, I used the brain as an inspiration to build AI models, to learn more about the brain, and use the knowledge that I learned from that to build even better models. So, this is the overview of the work. First, visual information is entering the brain, and then from that we have data. The data we use then to build a computer model that is able to capture these patterns that the brain cells carry, the information. And the goal is to basically learn how the brain represents the world around us by doing this brain reading technique. So first I'll talk about the visual information that goes into the brain. So when you look in front of you, all the information, like the space that you see that enters your eyes, is called your visual field or visual space. And this visual space, is just what is able to go into your eyes when you look in front of you. And in order to show you how the brain cells kind of work, I drew them here. And if I were to show you a black dot, on the top right corner of your visual field, then certain neurons or brain cells will fire or be activated. And if I change the location of the black dot, other brain cells will activate. So these brain cells are very spatially precise, or they are changing based on the spatial location of the input. And brain cells are also object specific. So not only do they carry this information about location, but also if I were to show you a tree, certain brain cells will be activated as well, or a mushroom. So this what and where of what the brain cells are responding to, that is called the receptive field of that brain cell. And I made use of this concept of receptive field to build my computer models and make them better, which I will talk about a bit later on. So I need brain data to build these models, right? So I have human data and monkey data. For human data, a guy was laying in the brain scanner while watching Doctor Who for 24 hours. And for the monkey had a little brain implant, and it was looking at different images. So now I will talk about the computer vision model that transforms these brain signals into pictures. So the computer vision model takes in brain signals, and these brain signals were being recorded while visual processing was happening. So the guy, remember, was watching Doctor Who, and these brain cells, they contain the information that about what the guy saw in the brain scanner, for example. And I used this computer vision model to predict what exactly the guy saw in the brain scanner. And I did not throw in the raw brain signals into the computer model, but I transformed this brain signal into a two-dimensional map that looks like a picture of a brain in 2 dimensions. And then I threw that into the model, which made the model better because it understood the information easier. Because you reduce noise in the input data, and you also give the model some spatial information as well. So yeah, this computer is able to read the brain. And these are the results of the fMRI, the guy that was watching Doctor Who. So in the top, you see the movie that he was watching, and in the bottom is the reading from the brain. And this is for the monkey. So for example, the monkey saw a picture of grapes, and in the bottom you see that the computer model was able to extract grapes from the brain. And with this model, I also compared it with older methods that existed in the field. So for example, the older method would show something blurry, while our method showed something that resembles more like a horse. And neurons or brain cells are organized in a very specific way. And for example, they are grouped in different brain regions. And V1 means early brain regions, and V4 and IT are later brain regions. So the signal, the visual information, first goes to V1 and then gets sent to other layers. And we can play around with that and see how each brain region carries what information, visual information. So in V1 here you see that it's simple color, V4 has more texture, and IT has more like complex conceptual, a person wearing something, and together all three of these form the final reconstruction, which is a t-shirt, a white t-shirt. And what we can also do is dive into the model, the computer model. The computer model has similar elements as the brain, so it has receptive fields, and it also has layers, which is inspired by the brain. And if we put our focus on these biological elements, we can also make the computer more efficient. So that's what I did, and it also, it made the model efficient, but it also allowed us to have more insight into the model because we can visualize these learning processes as well. So for example, here you see where the computer basically looked at. So to conclude, I managed to build a model that captured these patterns inside of the brain to reconstruct images properly that the subject saw originally, and I can use this model to test hypotheses about the brain. Overall, I built computer models that visualized brain representations and made AI models more efficient. So that's it.
Thank you for this clear summary, and we will now proceed with the defense of your dissertation. Before I give the floor to the first opponent, I would like to introduce the two people sitting next to me. This is promoter Professor van Gerven and co-promoter Dr. Gütschü. I'm sorry for the pronunciation. They already spent the last 4 years debating about the content of the thesis, so they will, in that sense, have done their job, and they can just sit back and relax for the next 50 minutes. So I would like to give the floor now to Dr. Vink from the Donders Institute for Brain Cognition and Behavior at our Radboud University.
Yeah, dear esteemed candidate, I want to congratulate you and your supervisors with this wonderful work, which I think is very important, it's timely, and it was technically also very impressive, your work. So I have a few questions, primarily on chapter 3, which is the chapter in which called Monkey See, and you analyze monkey data there to decode images from neuroactivity. And I'd like to exchange some thoughts with you on the interpretation of your findings with respect to the different areas and maybe also theories of vision. So perhaps starting a bit high level, there are these two classes of theories of vision. One is more discriminative, so the idea that the ventral stream extracts, let's say, categorical information in the higher levels of the ventral stream, like IT. And the other one are more classes of generative models, such as predictive coding, where you would think that the higher areas have latent variables that allow you to also reconstruct activity, right? And I was wondering, could you reflect if your findings on IT are maybe more in line with one or the other, or perhaps there are some results you have that could shine light on this question?
So, yeah, I managed to build a model that captured these patterns inside of the brain to reconstruct images properly that the subject saw originally. And what we can also do is dive into the model, the computer model. Dear opponent, thank you for your compliments. And so, to clarify, do you mean specifically IT? How IT hadn't showed like the results of the IT neurons?
That's correct. So, for example, you show in your figures that the reconstruction from IT is actually quite detailed, and I found it quite striking.
Yes.
So,
yes, I would not. I have not thought about it being related to predictive coding because I wanted to test that by incorporating more of temporal learning components in order to focus more on that. If I were to claim predictive coding elements, but what I speculated was that signals in the V1 and the V4 are propagating information from V1 to V4 and then to the IT. So after multiple dimensions and different features communicating together, or neurons communicating together to carry these features, they can become more complex. So that could be one of the reasons.
Maybe to follow up, you nicely show that V1 and V4 and IT do quite comparable in terms of reconstructing the image, right? There are some differences, but they're not so large if you look at the specific performance, right? So does it mean that IT actually has a representation maybe of the image that really allows you to go back to the original image, or would you actually say that no, it's encoding more categorical, sort of very high level features?
Yes.
Because,
for some examples, I think it's really difficult to evaluate with some statistics method. So I use my eyes to observe these reconstructions, and looking at the T-shirt, for example, you see that it's not a white. The final reconstruction and the monkey saw a white T-shirt, but in the IT there was a blue, even like person shape wearing a piece of clothing. So that made me think that the model needed all three brain regions in order to make that reconstruction.
Okay,
and
thanks.
Not just the IT.
Maybe to follow up on that, so you show that if you combine the three areas, you do a lot better in reconstructing the image than if you take the areas alone,
Yes.
right? And I wondered if that is a consequence of having more neurons in your model, so you have more data, or does it actually reflect some truly synergistic effect that the areas have more information together? And did you test for this in some way? And if not, how would you test for that?
So the amount of electrodes tells us basically how many signals are being picked up. So in V1, there were a significantly more amount of electrodes placed than V4 and IT. And even then, in V1, only simple reconstructions, shapes were being shown. So I think that already tells us that, the balance, the reason why there's more crispier reconstructions in IT is not because of the amount of electrodes, but it could be, just the right placement. We can never know.
What I mean is more if you had a V1 and you add V4 and you add IT, then you also have more neurons that go in your model, right?
Yes.
So if you would, let's say, take the same number of neurons from V1,
Yes.
do you think the model that mixes V1, V4 and IT does substantially better than having the same amount of signals from V1?
Yes, I do. So when I looked at V1 throughout the data set, there was consistently shown representations of color. So most of the colors that were being carried was in V1 neurons. So I would speculate if we had only V1 neuronal data, it will not give us better reconstructions, but more colorful reconstructions, but probably not the right shapes or the right object that we would be searching for, because that's what we've seen so far in the reconstructions.
Thanks. Maybe following up on that, with respect to training your neural network, you use a loss function,
Yes.
which was a sum of the pixel loss, adversarial loss, and a sum of the feature losses in a VGG 19.
Yes.
And I sort of wondered why you chose the loss as such, and for example, why you did not choose a perceptual similarity measure, something like LPIPS, which is validated on perceptual similarity judgments. And I wonder if this measure you have, did you look at perceptual similarity of human judgments?
Yes. So the reason why I used VGG L2 and adversarial loss is because it gives us a balance of the perceptual loss without needing a human to look at it, and also pixel loss, which is just more distance, and the adversarial loss for realness of the image, anything that would guide the model to go towards more of a realistic form of images. I have not considered the suggestion that you proposed because I was not aware of that one, to be honest. I did not think of also including human losses, because I thought it would suffice to just use perceptual loss of a neural network.
So maybe a last question on that. So, for example, with perceptual similarity judgment, it has been shown that it's the more deep layers that actually affect our perceptual similarity judgments the most, right? So not the early layers. So given that, what kind of measure would you actually choose? And do you think that also perhaps matches with the data that you have, the results on the monkey analyses?
Yes, I think that it would be a very good idea to adjust the weights of the losses based on the brain region that you're trying to reconstruct. I think that would be a good idea. Also, maybe make the weights of the loss and how they contribute to the training. Even learnable, like a learnable component, so that you don't have to make biases about that or bias decisions about that either. That's a good idea. Thank you.
Great. Thanks for your responses.
Okay. Thank you. The defense will be continued by Professor Battaglia, also from the Donders Institute here at the Radboud University, and he joined us today online. The floor is yours.
Yeah, thank you, Rector. Dear candidates, I would like to join Dr. Fink in congratulating you with very important and impressive work in your thesis. I can see that there are many repercussions for the clinical field, for the artificial intelligence field, for the brain-machine interface field. However, as a neuroscientist, I would like to ask you about what your work tells us about the brain. And before we go on, I have a small follow-up question on Dr. Fink's question. So still looking at chapter 3, and looking at the differences in so what you get from the different brain areas. And so I was also impressed by what you get from IT. And my question is, for example, in comparison with LIB1, and my question is, can it be that this is because, of course, you are training this network on a relatively small data set of images? So then essentially IT, because of its categorical and object selectivity, may actually fall back on one image in particular, and that has a chance to be the right one. Whereas V1 would work more pixel by pixel, and therefore being so under this situation suffer in the comparison. Would you think that if you had a much bigger data sets, the difference between these brain areas would sort of level up?
Dear opponent, I will think about this question. So I think the data set is actually quite large and diverse because we chose the things data set that contained a lot of variety and was made especially for avoiding the model to be biased on certain categories like dogs and cats of other data sets that occur quite often. Yes.
Okay, so that's not the reason why you think.
I think it's never a perfect data set, and it's a very a very data set dependent method. It's a very data driven way to do things. So I think it's definitely a good point to try to improve any way more data sets that represents the world and better. Yes.
Nice. So going to other aspects. So the other thing that you do that is very interesting for a neurophysiologist is looking at the timing. And so I'm talking in particular figure 3.3. So where you do your occlusion tests and show which time window do better at reconstructing the images. Of course, you show that if you use a larger time window, you get a better reconstruction. But so the thing here, so I guess figure 3.3 includes data from all brain areas, right? So we know that...
To be sure, yes.
So we know that, for example, V1 in comparison with IT, we would expect different latencies in the response.
Yes.
So in using the same small time window for all brain areas, aren't you penalizing your model too much?
Yes, we do have to make assumptions at some point of which time windows to select for which brain region. I did that based on literature, knowing how fast neurons signals travel. But it would be more ideal, definitely, and which is something I would have done if I had more time, to add a learnable component as well. So for the spatial locations, the model learns where to take the neuronal signal. But for the temporal dimension, that I did not implement yet. But it was, it will add a lot of complexity to the model, which is also a lot of computational power, but
But do you think it's fair to say that if you take a very early time window, so your response will be dominated, for example, by V1, whereas if you take a later time window, you would be dominated, for example, by IT?
Yeah, I think it can definitely have an impact on which brain regions will be used. Based on the time window.
And again, so another thing that was quite striking from a neurophysiology point of view. So in this, so you, of course, you also learn the receptive fields of your neurons. I'm talking in particular about figure 3.7. So what is interesting is that, so as expected, you get larger place fields as you move to the periphery of the visual field, and that is like the textbook.
Yes.
What is less like the textbook is that you get a lot of small receptive fields also in the periphery and also in IT. So there is a little bit less like classic neurophysiology data. So why this is?
Yes, well, there are a lot of V1 neurons present, but the ones that are not V1 neurons are also small. It could be an implementation issue because the way the network learns these circles is just learning a function, a Gaussian function, and I'm not entirely sure if that is the best way to do it. It could be that the model has trouble understanding.
So it serves the purposes of your model, let's say, but it's not per se a measure of receptive field size.
No, not yet. No, but I think it is. I think it is a step towards making something that can be a end-to-end model that also, while reconstructing, also gives you receptive field. But the focus needs to be on that, improving that.
So maybe I think I only have a couple of minutes left. So I would like to ask you like a more general question. So you work with both fMRI data in humans and MUAs. So can you compare like the challenges in working with these two different data sets? And so what?
Yeah, for me, if I were to, I'm on the modeling side, the challenges lie more in fMRI data because it's less precise, but it is a non-invasive technique, so I can imagine. So the challenges are dependent on who is working on it. But for me, the monkey data, electrophysiology, is very precise spatially and temporally. So that gives us a lot of insight and allows us to play around with the dimensions more in detail.
So what do you have to do different to squeeze information from fMRI data?
To do differently? Sorry?
Yeah. To squeeze information from this less precise fMRI.
So spatially, fMRI is quite informative, but temporally, I think it would be interesting to understand the delay of these signals better, and how exactly that the blood travels instead of recording directly.
Okay, I think I can stop here so I can give the floor back to the rector. Thank you very much for your answers.
Thank you.
Thank you. I would like to give the word to Dr. Herr from Maastricht University. Also, thank you for coming here from the southern part.
Thank you, Rector. Esteemed candidate, let me congratulate you on this beautiful thesis and, of course, also to your supervisors. I really enjoyed reading this thesis. It's a bit outside of my field, but I think I learned a lot. And I would also mention how impressed I was by your visuals in your thesis. Like in your presentation, you pay a lot of attention to detail, and this really shows. So, also very, very nice presentation. After the nice words, I would like to start very critical. Rather recently, Shirakawa published a paper criticizing a lot of the results in decoding from visual areas, reconstructing images, because he said many of the results are based on the corpus characteristics. And I think I could take some of these arguments and apply them to your Doctor Who data set, because it's all the same show, it's all the same monsters, as we saw in the presentation. So how would you defend this criticism?
Thank you, dear opponent. Thank you for your compliments, and I would like to answer your question. I also read that paper. I think it's a really nice paper, and they make really good points. They focus mainly on reconstruction models that take latent spaces from pre-trained networks, and the nice thing about my model is that it doesn't do that. So that already takes away a lot of biases from other data sets and the need to evaluate better that it's not biased by other data sets. But they did mention that the zero-shot method would be a good method to evaluate the model. And there is it's very difficult to find nowadays a data set that contains purely zero-shot images in a test set. So it would be really nice if we can find something, and probably we can then just test it on a zero-shot image that the network has not seen. So it could be that the model currently reconstructs, for example, that horse, and it should be a zero-shot image because that particular image was not seen by the training model during training. Also with Doctor Who, there was a splitting of test and training, and with different seasons and different characters, but it's still maybe the same concept. So yeah, to find something truly zero-shot is quite difficult to decide what, to set the threshold, like what is actually, and if the monkey has already seen it before, for example, or the human, does that still count as zero-shot? I don't know. Yeah.
Okay, thank you. I think that was rather convincing. Now, both data sets you used in your thesis were of one individual, and we always strive for generalizable models. How do you think your models would fare if we put another person in the scanner for 24 hours watching Doctor Who? Do you think the model would be transferable?
This current model, I don't think so, because it was not built for that. I am not really, to be honest, I was not really focusing on that. I was more focusing on diving into the model and the processes. I think it would not work if I put someone else's brain signals in there. We did try to, in the beginning, there's also for this person some kind of imagery signal, so it's the same brain but different types of signals, and that also was very difficult already. But
I agree. So the question is, what's necessary to build a generalizable model? Do you need a lot of data of a few participants, or do you need a lot of participants with rather few data in each of them?
I think that's very difficult to answer for me.
And that's why I'm asking you.
Yes, thank you. I think the important part for the model to know is the receptive fields, the mapping, because that is that already kind of aligns the brain with each other if the receptive fields are the same. But in order to achieve that, you would probably need a lot of data from different subjects and not necessarily a lot of data from one person.
Good.
Relative.
Let's try that next. I come from the brain-computer interface world, and first I want to hear, how can I use your results to build a brain-computer interface?
So for a brain-computer interface, you would need for vision at least, you would need a very precise mapping between whatever is going to reconstruct and the locations of where the neurons are being induced and what will come out of it. So my method is very good at reconstructing this or finding this relationship between space in the brain and space in the visual field. Mm-hmm. So I think in order to implement something that requires this precise mapping, you can look at the model and play around with it before you dive straight into the brain. Yeah, it can be a tool to understand the relationship more precisely between space and also features.
Okay. I saw that in the monkey data, at least, you averaged over 30 single trials.
For testing.
In the testing.
Yes.
For a BCI, of course, we would want to do this with a single trial.
Okay. Yes.
What's necessary to achieve the same quality of results in single trial?
So I think that is also something that is a similar question as the zero-shot learning. So if we have a repetition of something, then it becomes cleaner and less zero-shot. So if we build our model with this evaluation metric that the recent paper suggested, I think that would come closer to being able to reconstruct something without needing repetitions. I hope.
And have you tried?
No, I have not tried using non-repetitive, but I think that is a good point to try, because that's, I do agree with the authors saying that that is one of a really important evaluation metric if you were to claim that you're actually making brain reconstructions.
Do I have time for one more? Good, then I want to ask a more technical question about the monkey data. If I understand the pre-processing correctly, you're using 2 frequency bands, but you're not looking explicitly at firing rates of the multi-unit activities. Why so? Why not using firing rates?
Well, it's not necessarily that I didn't want to do it. I just was very excited to use the data that I got, which is just the envelope.
Mm-hmm.
But I was very interested in the spatial information, and if I were to continue studying these communications between neurons, I would also be interested in temporal information. So then that would probably, be the time when I will look at the firing rates.
Looking forward to that. Thank you very much.
Thank you.
Thank you. I would now like to give the floor to Professor Cornelissen, who is from the University Medical Center of Groningen. So big thanks for being able to join us today.
Thank you, Rector. Dear candidate, I also like to congratulate you and your supervisors with a very nice and a very interesting thesis. As you may know, my background is in visual neuroscience and ophthalmology, more than in machine learning. So I've read your thesis with a lens that reflects this background. There was an intended pun there, because I'll come back to lens. You've taken some well-known properties from neuroscience and applied these to do improve your reconstructions. Like one of these is retinopathy, which you do in chapter 2. Then you estimate receptive fields and apply these receptive fields to your reconstruction models. I hope I got that right, approximately. Yeah? But then I read at the bottom of page 25 that you transformed your images using a fish eye or a fish lens transformation. So that's where the lens came in, to mimic biological retinal eccentricity. My gut feeling is that you are referring to the fact that retinal resolution declines with eccentricity, and that you somehow wanted to compensate for that. But in a way, I got a bit lost here, and I'll tell you why. Because I think your retinotopic modeling would already take that into account. And so my first question is it possible that by doing this transformation and doing the retinopathy, you sort of overcompensated for these eccentricity effects?
Yes. Okay. Dear opponent, thank you for the compliments. I do agree, and that's also the reason why in the later chapters I also did not apply this this filter, let's say. But when I applied it on the first chapter, I was reading a paper that basically convinced me that I should do it. And and I tried it out, and it looked like, something that would make sense. It just at that point made sense. And also at that point, I did not add any learnable components in the receptive field yet. So it was purely relying on the spatial location of the receptive field. And I did not assume that would also already take the warping, the resolution downsampling with it. But later on, I did remove it because the receptive fields were being learned. So, yes, that.
Okay, so I'm glad I understood this correctly. But you apparently did you ever try to redo it without this transformation to see if this improved or your results even further?
Yes, I did redo it and it was the same. So that's why I.
Right. Yeah. Because in some of these pictures, it seemed like there was a bit of a zooming in on the center of the image in the reconstruction, like some enlargement. I'm not sure if that's related to it, or whether that's just another property of the human brain data.
In the reconstruction.
Some of the examples I didn't write down the exact figure numbers, but you showed a couple of examples of reconstructions and. Yeah, occasionally it looked like, there was a zooming in on. So
the reconstructions are basically guided by the targets or by applying this filter on the target frames, so the original frames, we're making the assumption that the brain receives this information with that filter. So the reconstruction, when it gives us that warped image, then the assumption is this is how the brain represents this reconstruction, or sees this reconstruction. So it's not like how we see it, but more like the brain before our eyes or after our eyes.
So in a way, you should have unwarped it to make it more like see it, right?
Yes.
Okay. All right. Okay. I want to stick with the retinotopy concept because you applied it to your models. But I was wondering, there's many aspects to retinotopy. One is this eccentricity scaling, so to speak. And I was wondering, how did your reconstruction models exactly benefit from this retinotopic scaling? And as a follow-up question, can we learn anything about the human brain that we didn't know yet from your reconstruction models about this retinotopic scaling?
Yes. So this model escalated. It started with just learning the receptive field, which is a well-known phenomena. We can't really claim that we learned something new from receptive fields, but it was kind of an indication that this tool is reliable and can be, worked on to build something more complex that will tell us eventually something new. And so in the last chapter, we added these attention components that also resembles receptive fields, but has a higher dimension of receptive fields. So in this way of learning receptive fields, you can visualize, basically, I don't know how else you would be able to visualize this, but the differences between spatial receptive fields and feature receptive fields. So not only can you see the receptive field of that particular neuron, but you can also see how much it activates per feature. So the spatial receptive fields were really consistent with across the, for example, the V1 neurons. They were all, what you would assume a V1 neuron spatial receptive field would look like. But if you look at the feature receptive fields of V1 neuron, they all vary. And it's basically a new method to show that it's possible to disentangle these two dimensions
So you mean the spatial location and the feature,
Yes.
so the spatial dimension and the feature
Yes,
dimension?
yes.
Yeah.
So how do these neurons respond based on features, but also location, and not just location?
So is it based on that you can predict that you would expect certain receptive field properties, say in V1, that are currently not known from human from neurophysiology, monkey neurophysiology, or human retinotopic experiments?
So I did
That's
not.
what I'm a bit looking for, right?
Yes.
What can I take home and study in my future human research experiment?
So it's what I think that is quite fun to think about is that the model is a tool to play around with in these dimensions. So it's not that we can do that with the real brain, and it's something that I think would be really exciting if we can make a system that can tell us about all these dimensions and how, where we can change where we can zoom in to these different dimensions, what the model is trying to tell us that it uses to learn these reconstructions.
Okay, so you think also in the human brain that kind of learning would take place? Is that what you're saying?
I'm not saying that I think that's the case, but I think it's a nice way to study whether that is the case. Okay. And it is a nice tool to use in addition to tools that we already have, but also it just gives us a very different perspective on how to understand these processes better.
Okay. Yeah. All right, I'd like to continue with chapter 5, at least the concept that you use there, this inverse receptive field, and you use it in a way also to understand attention or to see how attention affects processing. Again, if I got it right, because again, I'm not a machine learning person, so I try to read between the lines mostly. And there again, I thought, okay, this is a really intriguing concept, but unfortunately, and then I had hoped to read something in your discussion or so about, so what can we do more with this, right? Can we, well, I didn't come across anything clearly that said, okay, we can apply it to this in this way. For example, in humans. So my first question is, because you applied it to the monkey data, right, I believe, and can we also apply this concept in fMRI experiments? Would that be possible in human fMRI experiments?
I think it definitely is possible because the receptive fields, the inverse receptive fields, are very spatially, dependent on spatial precision of the data, and that is, fMRI is quite spatially accurate. The thing that I think is also interesting is to look at the temporal dimension, and I think with fMRI that is less likely to be able to do that. But that is something we, in the discussion, I don't know if I mentioned it, but that is something that would be a nice next step to add temporal dimension, not just spatial receptive fields and feature receptive fields, but also the other dimension that's also important. And by looking at the entirety of this communication between neurons, we can definitely see a lot of, gain a lot of knowledge about that. But I don't dare to make, neuroscientific conclusions from these maps yet. But I think it would be really nice to be able to do that eventually. Yeah.
But I still want to, if I have some time.
A very, very short question.
A little
or
challenge.
Short answer.
Whether you could think of any medical application that you could use this particular inverse receptive field attention modulation.
Yes.
For.
So,
have you thought about that?
The reason why we have this data is because there's a big group of researchers working on cortical implants for blind people, and in order to properly stimulate perception, induce perception, you also need to understand the receptive field quite accurately. And I think in order to, if you were to take the brain signals of a person, it's very individually focused. I think that's important to do if you're making an implant for someone, because it's tailored to that person and that person's brain. And yeah, this method allows you to do that, to train something that is single subject. You don't need more data from other people. And yeah, it's a good way to focus on one brain and less biases from other data sets.
Okay, thank you. I had some other ideas I wanted to test with you, but I don't think I have the time.
I'm sure there
So
will be
thank
an
you very much.
opportunity
Yeah.
later today. I now give the word to Dr. Dijkstra from University College London. Also, thank you for coming here today.
Thank you, Rector. Esteemed candidate, I would like to join everybody in congratulating you and your supervisor on a really good thesis. I really enjoyed reading it. I thought it was really cool to also read your introduction about how biology has inspired AI, and I think that your thesis itself is a really nice example of doing that as well, with including retinopathy in AI models. So well done, very cool to read. It was also really nice to see what Rowan's brain data was being used for after all these years. So you mentioned before that there was also an imagery task, and you tried your models on the imagery data, and you said that it didn't work that well. Could you explain a little bit about what you would need in your models to be able to reconstruct imagery?
Thank you, dear opponent. Yes, so I have studied the visual cortex about with regard to visual input and mental imagery. I have not really, dived into that field a lot, but I can imagine that you would need very strong signals of mental imagery. So one thing that I can think of is a person that is very good at mental imagery. So not just a person who watched Doctor Who for 24 hours. But in that sense, I also think that after watching Doctor Who for 24 hours, I could imagine them being able to visualize something very well. So yeah, building a model around visual imagery would probably also require different types of training data, because yeah, I think maybe same data could be used, but some kind of transformation between the images and making an assumption of how visual imagery would look like instead of the actual input, using the actual input to train the model, but more of like a very blurry filter, something dreamy, and then kind of asking the subject, is this how your mental imagery looks like? Maybe that would be a technique.
Sounds like a cool new project. Great, thank you. Another question is that you focused really on the visual ventral stream, which makes sense because you're looking at visual perception. But there's quite some research showing the involvement of other brain regions also on visual perception. And there's even this recent stuff by Thomas Knape showing that even high-level brain regions also contain receptor fields and retinotopic organization. So do you think that adding more high-level brain regions to your model would improve reconstruction? And if so, how?
Well, yes, I do think it will add more information to the model, which always is a good, if it's a relevant information, then it's good. It might, would require us to change the model a bit based on, to align it better with the data. So currently it works with the brain data that I played around with, but I don't think it would have to change a lot, because it's, yeah, the technique, it basically takes, especially if it already has receptive fields, then it will take the data as an entirety and also the receptive fields, and is able to capture information from everything. So I think it would not be needed to change a lot.
Nice, cool. Okay, the other question is kind of related to my previous my colleague's previous question on the attention model. So if I understood that model correctly, which I'm sure I didn't entirely, but it kind of read to me like a bottom-up attention model where kind of features are selected to pay attention to, but it's not something that changes depending on the input. Whereas I guess in the brain, you can employ attention depending on your input, so you can flexibly change which features you attend to. So is there some way of implementing this more top-down version of attention in your neural network?
Yes, so the attention mechanism refers to where the model pays attention to in the input space. So it's more the attention of the model. And the model is being guided by visual neurons and not attentional neurons. So I think one step towards modeling these top-down or bottom-up attention mechanisms, we would need data from that task specifically. What the attention modules in IRF tells us, the chapter, is just basically which input signal is being used to make that reconstruction in the end, and which spatial receptive field and which feature receptive field.
Okay, thank you. Do I have some time for? Okay, cool. So you're at the end of your PhD, you finished. If you had all the money and time in the world, what would be the next experiment that you would do?
All the money. Well, I think.
What is a research question that you would really like to answer?
Yeah, it's the same, research question. I've always been interested in learning how learning works. Mm-hmm. So it's not necessarily visual processing it's just, curiosity. I'm very curious about how everything works. So honestly, I think it's really fun to figure it out step by step, and different elements and different perspectives. And I don't have like a very strong opinion about how things should be done, as long as it's correct and scientifically sound. And I think in neural decoding is also a really fun way to approach things. Yeah, thank you.
The defense is hereby concluded. I give the floor once again to the candidate.
Having defended my dissertation to the best of my ability, I would like to thank you, Rector, and you, my supervisor and co-supervisors, and all of who have honored the ceremony with their presence.
The doctoral examination board retreats for its deliberations.
Okay, we're allowed to talk. Ze komen zo terug.
Dat is heel veel apart.
On behalf of the Doctorate Board of Radboud University Nijmegen, we have decided to award you the degree of Doctor. I invite Professor van Gerven to discharge the task assigned to him.
With the power entrusted by law to the Doctorate Board, I hereby confer upon you, Lynn Lee, born in Harderwijk, the title of Doctor from Radboud University Nijmegen, to which are attached all the associated legal and customary rights and duties with respect to academia and society. As proof thereof, I present you with the doctoral diploma signed by the rector and the doctoral thesis supervisors and co-supervisors.
The co-promoter, Dr. Güçlü Türk, will now address you in the laudatio. You may be seated.
Dear Lynn, dear Dr. Lee, I want to start by telling you how Lynn and I first met, because it really captures who she is. It was, I think, 2018 or 2017. We were at a consortium meeting in Amsterdam. I had just started my postdoc position in Richard van Wezel's lab, and my project was about building algorithms for visual cortical implants, an emerging technology still under development today. Suddenly, this bright-eyed medical biology student from Radboud University approached me and said, "Hello, I'm Lynn, I'm your student." I was completely confused. You see, she had initially talked to Richard about doing a project with him, and he hadn't exactly mentioned her to me yet. So, yeah, but that is Lynn. She's decisive, direct, and already 2 steps ahead. She ended up writing her thesis with me, working on a project evaluating biological motion perception for visual cortical implants. Then she did a literature review study with Umut, who would later become her main PhD supervisor. Along with a few more projects, Umut wrote a grant proposal for Lynn's PhD and submitted it to the donor's internal PhD round. He was awarded funding for all of the submissions he was involved in. Well, that's not surprising because he's a brilliant scientist, and more importantly, because I helped him write them as well. The next year, they changed the procedure to prevent him from sweeping all the grants ever again. And that's how Lynn joined the neural coding lab of Umut, her main supervisor. He was so exceptionally productive and selflessly supportive of his students at the expense of his long-term well-being. Without ever receiving the support he clearly needed. Yeah, this part is a bit emotional. And I'm deeply sad that his health doesn't even allow him to join us online today to celebrate this moment with us. Lynn, it's difficult to fully celebrate your achievements without celebrating what you and Umut accomplished together. But we'll do our best. Eventually, we started having joint lab meetings, my neural engineering lab and Umut's neural coding lab together. Those meetings were the highlight of my week, because the excitement, the progress, all those sharp minds like Lynn, it was electric. Except for my own long-term sick leave right after COVID period, I had the privilege of witnessing Lynn's development into a bright, independent scientist that she is today. Now, Lynn, you are remarkably multidimensional, and I need to talk about what it has been like having you in our labs. So you love science, we all know that so far, and you always have. But I believe even more than that, you love dogs. I once told you, I really love dogs, but I've never had one, so I don't know how to interact with them. And you said with complete certainty, there's nothing to know, you just love them and they love you back. Your Banoi had dogs. I hope I said it correctly. Your grandma, that means. And you loved playing with them. With your encouragement, I ended up getting my own dog, Buffy. So to give you a sense of Lynn's commitment to this mission, when I asked if she would be willing to visit Buffy as a puppy before I decided whether I would be getting her, she didn't even hesitate. She took me to Tilburg to meet Buffy for the first time. A few weeks later, when I decided I had to have her, Lynn drove me there again. We picked up this tiny puppy and we drove back to Nijmegen. Buffy was very vocal about her discontentment with the car ride, and Lynn got us home all safely despite all her cries. Then she even dog sat when I needed help, and she dog sat for others too. And I think she's still doing it, because just last week you mentioned picking up a dog, didn't you? Yes. So then there's bouldering. So I mentioned you are multidimensional, so I cannot not mention bouldering. So Lynn started bouldering during her PhD and hasn't stopped since. She has gone on trips to France to experience the best climbing locations in nature. She became quite an expert, even created an Instagram account called Lynn Boulders. And her last post says, calming my nerves with 1 hour tension boarding because tomorrow I have my PhD defense. I hope that helped, Lynn. She also has thousands of followers, and she even runs her own business making specialty care soaps for hands stressed from bouldering and chalk. And I also have to mention gaming. Lynn got a Nintendo Switch during her PhD, and I remember her and Katya enthusiastically discussing Zelda. And true to Neural Coding Lab spirit, today we saw her presentation adorned with pixel art and game references, with Lynn herself as the main character navigating through her research. All while publishing at top conferences like NeurIPS and CCN, of course. Lynn, you are surrounded by people that love you and you love. Your grandmother, your brother, the rest of your family, and your boyfriend, Noor. And now you are starting an exciting position at the Netherlands Forensics Institute, where you'll bring your brilliant mind to solving mysteries and fighting crime. At least that is what I imagine you'll be doing. So let me close by saying, Chuk Mung Za Ding. I tried to say congratulations to the family, some of whom flew here all the way from Boston. And Lynn, congratulations to you. We are so, so proud of you.
Thank you, co-promoter, for these very nice words. I think we learned a lot today also about visual experiences, but also Zelda, dogs, and bouldering. And Doctor Who, not to forget. I would also, on behalf of the doctorate board, congratulate you and your family, your beloved ones, with this massive achievement, this great achievement. So congratulations. And before I close the ceremony, I would like to give Professor Battaglia once the opportunity to congratulate you.
Dear Lynn, thank you so much for this defense. It was a pleasure to read your thesis, be involved in your graduation, and I wish you all the best for your future work and all your activities, and hopefully see you somewhere. My apologies for not being there in person. I'm currently on sabbatical. That's the reason.
Thank you.
Thank you. So we have now come to the conclusion of the ceremony, and ask you all to stand. Oh, wacht even, nog niet, hè? Niet volgens mij. zelfs. We close with the following words. Gratias tibi agimus omnipotens Deus, pro omnibus beneficiis tuis, qui vivis et regnas per omnia saecula saeculorum.